Good day, students. In today's lecture, we are going to talk about the single membrane bound organelles like the lysosome, peroxisomes, and glyoxysomes. In the previous lectures, we have discussed the other single membrane bound organelles like vacuoles, endoplasmic reticulum, Golgi bodies, and the double membrane bound organelles like the plastids, mitochondria, and other part of the cell. So in today's lecture, we are going to start with the single membrane bound organelle, that is the lysosomes. Now, first of all, regarding their discovery, the lysosomes were discovered by Christian de Duve. If we break the word lysosome into two parts, it makes lyso, that means digestive, and soma, that means body. So lysosomes are the digestive bodies or the structures. So the basic function with which the lysosomes are related are the digestive functions, because the lysosomes contain hydrolyzing enzymes which are commonly known as the acid hydrolases. This clearly means the pH of the lysosome is acidic. The lysosomes are also known as commonly as the suicidal bags. It is regarding their function during which they can digest or degrade the worn out or unnecessary organelles of the cell itself. Or even in some cases they can degrade or digest whole of the cell itself. Therefore they are commonly called as the suicidal bags. Regarding their presence, they are mainly present or found in the animal cells, but they also occur in the plant cells, although few in number. So we can say that they are found mainly in the animal cells. Now coming to their function, as I told you earlier that they contain the digestive enzymes or the hydrolyzing enzymes. So their main function is related to digestion or degradation or breakdown. That's why they are also known as the digestive body or the suicidal bags. So their first function is to digest the liquid or solid materials or molecules taken by the cell by the process of phagocytosis and penocytosis respectively. And this whole process of digestion of the external material taken in by cell is called as heterophagy. So I'll explain once again what is penocytosis. Penocytosis is taking inside of the liquid droplets by the cell whereas the phagocytosis is the intake of solid particles by the cell. And when the cell digests such solid or liquid material taken in, this process is called as heterophagy. The second important function of the lysosome is autophagy. Autophagy means self-lysis. Phagy means lysis. Auto means self-lysis, whereas heterophagy means lysis of the external or foreign particles. So, in autophagy, the lysosomes digest the worn out cell organelles or unnecessary part of the cell or even the whole cell when required. So this is all about the cell organelle, that is lysosomes. Now the next cell organelle, that is single membrane bound cell organelle, is peroxisomes. Remember that lysosome is also a single membrane bound organelle. Now we are going to talk about the peroxisomes. First of all, let us discuss who discovered peroxisomes. They were firstly discovered by Rodin in the liver and kidney cells of the animals, whereas in case of plant cell, it was discovered by Talbot in the germinating seeds. The peroxisomes are small ovoid single membrane bound sac-like structures. The peroxisomes contain the enzymes which are known as the oxidase enzymes. So we can say they contain Now regarding their function, they perform different functions in the animal cells and the plant cells. For example, we can say that in animal cells they carry out lipid metabolism, but in case of the plant cells they carry out photorespiration. Photorespiration is a special process which takes place only in the plants which carry out photosynthesis by the C3 mechanism that you will be studying in details in the higher classes. Photosynthesis, you all know, is the process of fixing the carbon dioxide which takes place only in the presence of sunlight in the green cells of the plant which contain chlorophyll as the pigment. But some of these plants, they carry out a process, a modified process in the presence of high sunlight, high intensity of sunlight or high oxygen concentration. Instead of fixing up the carbon dioxide, they start releasing the carbon dioxide, and this process is called as photorespiration. It is different from the normal respiration process. It is something you can say is the modified process of modified type of photosynthesis mechanism, in which instead of CO2 being fixed up, it is being released. Now, after peroxisomes, the next or the last single membrane bound organelle that we have to discuss is the glyoxysomes. So let us discuss it. The glyoxysomes were discovered by Beavers in 1961. And regarding their presence, they are found mainly in the plant cells and particularly in the germinating fatty seeds, that is the seeds which have high amount of reserve in the form of lipids or the fats, like resinous seeds. Now coming to their function. The main function related to glyoxysomes is that they convert the fats to carbohydrates by the glyoxylate cycles. Glyoxylate cycle is the mechanism by which the fats are converted into the carbohydrates. Now let us discuss about the non-membrane bound organelles, and that is example is the ribosomes. Ribosomes were discovered by or reported by Robinson and Brown in animal cells. They are the smallest cell organelles without any membrane covering membrane, and they are spheroidal in shape. Regarding their composition, the ribosomes are ribonucleoproteins, which means they are made up of protein plus the RNA, that is ribonucleic acid. The RNA, that is ribonucleic acid or ribose nucleic acid, is rRNA types, that is ribosomal RNA. RNA stands for the ribose nucleic acid. It is a type of nucleic acid or the genetic material. Besides rRNA, the ribosome is made up of proteins. Therefore, we call it as a ribonucleoprotein. They are of two different types on the basis of their presence in the prokaryotes or eukaryotes. For example, in eukaryotes, the ribosome is 70S type, whereas in eukaryotes, it is 80S type. This 70S ribosome in prokaryotes is further made up of two smaller subunits that are known as 30S and 50S. Out of these, the 30S is the smaller subunit, whereas the 50S is the larger subunit. Now, the second type of the ribosome is 80S type, which is found in the eukaryotic cells. Again, as in case of prokaryotes, in eukaryotes also the ribosome is made up of two subunits, and these subunits are known as 40S and 60S subunits, out of which the 40S is the smaller subunit and the 60S is the larger subunit. Now, what does this S stands for? The S stands for Seeberg constant or coefficient or Seeberg unit. And this Seeberg unit, or S represented by S, is used to measure the sedimentation coefficient rate of the ribosomal units or the ribosomes. Basically, when we subject these ribosomes to centrifuge, then they sediment at different speeds. So that speed is measured in terms of the Seeberg unit. Now coming to the functions. Their main function is that the ribosomes help in the protein synthesis. The process of protein synthesis is known as translation. So, the ribosomes participate in this translation process. So, this is all about the cell organelles. We have discussed different types of cell organelles. So, by now we are much familiar with the internal structure of a cell. Now, let us discuss the differences between a plant cell and an animal cell on the basis of the knowledge we have gained till now. So, we'll discuss the differences among the plant and animal cells. There are very striking differences present between the plant and the animal cells, although both are eukaryotic. Eukaryotic, you all know, that have a true nucleus, and a eukaryotic cell has, besides true nucleus, it has well membrane bound organelles like the mitochondria, Golgi bodies, endoplasmic reticulum, peroxisomes and plastids, etc. Now, if we discuss about the differences between plant cell and animal cell, the first major difference between the two is the presence or the absence of the cell wall. In case of plant cells, the cell wall is present, but in case of an animal cell, the cell wall is absent. Cell wall is the outermost boundary of the plant cell, whereas in case of animal cell, it is the cell membrane which forms the outermost boundary. In case of plant cell, the plastids are present. Chloroplast is one such example of the plastids. Other type of plastids include the chromoplast and the leucoplasts. Whereas in case of animal cells, the plastids are altogether absent. In case of plant cells, they have a single large vacuole which occupies 90% of the cell. But in case of animal cells, they have numerous small vacuoles. The last difference among the two is that in case of plant cells, the centrioles are absent, whereas in animal cells they are present. The main function of centriole in the animal cell, the centrioles are absent in the plant cell. The main function of the centriole in the animal cell is spindle formation during cell division. So this is all about the difference between the plant cell and animal cell.